Hello, everyone. Thanks for coming to my talk, which is titled A Holistic View of the Factors Shaping the Diversity of the Lichen-Forming Fungal Genus Sticta in the Caribbean. I hope you like it, and let's get started. So in 2008, Rick Lefts and Birmingham stated that the Caribbean islands, which I'm showing you here in orange, were an outstanding natural laboratory for studying processes that establish patterns in the diversity of life. And certainly, the Caribbean islands have been really important for both ecological and evolutionary studies. However, probably the most famous case study for this are the Caribbean anoles. So this amazing group of lizards have, as many of you know, have profusely diversified as a result of colonizing these islands. And this diversification is not only limited to the number of species, but also to an incredible set of distinct morphologies, I should say, that have independently evolved in each of these islands. So findings from this study have allowed us to understand important evolutionary processes, such as diversification in islands and adaptive radiation. But it remains to be seen how much of these findings apply to other important and biodiverse groups that have been less studied in the region, such as lichens. So lichen diversity in the Caribbean is really the knowledge is still really rudimentary, but some studies are beginning to change this scenario. And one of these I had the honor of leading. In this study, we perform a phylogenetic analysis of the fungal of the lichen genus Sticta, specifically in the island of Puerto Rico. And the idea here was characterizing the diversity of this group. But one of the things that we found is that the diversity of this genus and this island apparently had stronger biogeographic links to South America. Unfortunately, this remains as a testable hypothesis because we didn't carry out analysis for ancestral range reconstruction or diversification. So motivated by this study, the work I'm gonna present you here revolve around this important question. What are major ecological evolutionary factors shaping Caribbean Sticta diversity? Here I'm showing you one of the key species, Sticta damicornis, which is a presumed Caribbean endemic. And for this study, I followed a two-pronged approach. One, I use a macroevolutionary approach to characterize regional diversity, number of species in the region, also trying to understand a little bit more about the timing of colonization, when did Sticta arrive, and also from where the species came from. The second part of this study follows more of a community phylogenetic angle. And specifically, this part aimed at addressing relatedness between island-level communities, both at the taxonomic and also at the phylogenetic level. I was also interested in trying to understand the potential role of environmental filtering versus dispersal limitation to explain these patterns of relatedness. So the sampling for this study I'm showing you here in this map. These are the areas that I visited for this study, which I'm showing in colored dots. This includes all the islands in the Greater Antilles, but also a few of the islands in the Lesser Antilles. So from the samples I collected here, I generated data for 6 loci, and then I performed a candidate species delimitation analysis because we really know very little about the identity and diversity of this group in the region. So for this, I perform an integrative taxonomic approach, which followed more or less the approach that I did for this Puerto Rico Sticta study. So for the macroevolutionary angle of the study, I carried out the divergence dating. For this, I used the program BEAST and to generate my ultrametric tree. And then I used an alignment that included the candidate species from the Caribbean together with the previously delimited species from a broader and global Sticta sampling. I carried out also ancestral range reconstruction based on the dispersal and extinction collagenesis model, the DEC model from William Smith, which allowed me to reconstruct ranges in ancestral nodes in this ultrametric tree. And for this, I basically assigned each of the species analyzed to each of these 9 biogeographic regions that I'm showing you here to the right. So for the community phylogenetics part, I analyzed island-level communities, and first I generated a presence-absence matrix of the species stratified by island. For the analysis, I performed a beta diversity analysis. Specifically, I generated data for the Jacquard taxonomic dissimilarity index, and also for the UNIFRAC phylogenetic dissimilarity index, which is basically very similar to the Jacquard, but it adds a time component by using data from a phylogenetic tree. I also use Fossey-Siemens clustering to basically see if the species cluster more or less into distinct island level, I should say, clusters. And then I was also very interested in seeing if this patterns of dissimilarity as measured from this Jacquard and UNIFRAC indices correlated to one of two variables. First, environmental variables, which I basically obtained from remote sensing data. This multivariate data set I reduced with a PCA, and I used this data to generate an environmental distance matrix. And I also used I was also interested for addressing the dispersal limitation question. I generated a geographic distance matrix, and for this I use intra-island distances. So for the results, we uncovered 64 candidate species for this region. Interestingly, 59% of these are potentially endemic. I cannot show you all the species, unfortunately, but here is just a small sample of the incredible diversity of species and morphotypes that you find in this region. So for the ancestral range reconstruction analysis, here's the main ultrametric tree with the reconstructed ranges in each node, which I'm showing you here as pie charts with different colors. And the important colors to keep in mind here are the purple, which indicate Caribbean restricted species, this reddish color, which shows a broadly distributed species, basically from Caribbean and South America, and then the blue, deep blue color, which show species that are apparently restricted to South America. So all the species from the Caribbean were nested in the main clades A, B, and C. And I'm gonna point out a few of these nodes that are very important. So specifically A and B were reconstructed to be South American in terms of origin, and all the Caribbean species nested in these two clades basically derived from ancestors that came and colonized straight from South America, and either remain broadly distributed, and some of these, as some of the species here in clade B show, became restricted to the Caribbean. Node C is important as it shows the first instance of colonization of Sticta in this region, and this apparently occurred around 19 million years ago. Node D is within clade C, and it's important. As you can see, it shows a species with a restricted, an ancestor with a restricted Caribbean range, and that led to species that apparently expanded the range, in this case to Central America, as the green color shows. So this shows the first potential case of reverse colonization, which means species that are able to come back from islands and recolonize the continent. Node D is also very important. Similar to D, it shows Caribbean endemic ancestral species that apparently that led to species that apparently expanded the range to the Caribbean and either remain distributed just within the Caribbean. Some colonized back, as some of these nodes here suggest, and some of the species that actually colonize back the continent lost their distribution in the Caribbean, as this blue color suggests. So again, two potential cases of reverse colonization. For the clustering analysis, here are the results for the Jacquard and UNIFRAC, and I'm using dendrograms to show similarities between these island level communities. And at minimum number of clusters 2, we see a high similarity between Hispaniola and Jamaica for both indices. And interestingly, when we look at the minimum number of clusters of 3, we get more or less the same pattern from 2 clusters. However, we see a separation from Cuba and having then Puerto Rico and the Lesser Antilles in one extreme, and then Hispaniola and Jamaica in the other extreme. For the correlation analysis, the variables that explain most of the variation in the PCA were elevation and temperature, which are then used to explain these results. And as these plots show, we found that the environment apparently explained much more of the variation in this dissimilarity indices for both Jacquard and Unifrac, and these were statistically significant. However, when we look at geographic distance, we really couldn't find a strong association between these variables and the dissimilarity diversity, beta diversity indices. So as we saw, the Caribbean Sticta apparently has stronger links to South America. However, we need to take a closer look at Central America because the species in this region are undersampled in our study. And there's a chance that there's stronger connections to this region too, as other studies for other groups have shown. Reverse colonization is important to indicate too, because this sort of goes against theoretical odds. And this is predicated on this idea of island syndromes. Species that are apparently evolved within islands sometimes can lose their dispersal and competitive abilities, which preclude the probability of them colonizing back the continent. And our study shows evidence that perhaps this might be a little bit more common in lichens, and which is not limited to lichens, as this example with this moss that I'm showing you here evidences. For the community phylogenetics, a stronger association between environmental variables and beta diversity indicates that perhaps environmental filtering play a strong role in explaining the patterns of relatedness between these island level communities. We also saw affinities in terms of the composition of species in the Lesser Antilles and Puerto Rico. This has been previously shown for other groups, especially plants, but we need to be careful. Some other studies indicate that Puerto Rico might be more similar to the Greater Antilles. But for us, I want to propose the idea of hurricanes, as the maps in the right shows the most likely paths of hurricanes in the regions tend to connect more Puerto Rico to the Lesser Antilles. So there's a chance that hurricanes are probably connecting more these sets of islands as opposed to these islands to Hispaniola and Jamaica. Also, I want to highlight the strong differences between Puerto Rico and Lesser Antilles and Hispaniola and Jamaica. And a lot of this is probably explained by the presence of small range endemic species that are restricted to the high elevation environments of Hispaniola and Jamaica. So with that, I want to thank the IAL organizing committee, to my funding sources, which are shown in images here, and especially to the Field Museum Lichen Group and the Pritzker Lab. Thanks a lot to all for the support. And of course, none of this would have been done without the collaboration of important regional colleagues, which I cannot name here, but are properly thanked here. And to all of you for coming to my talk, thanks a lot. Have a good one.